All right, ladies and gentlemen, so welcome back. So we are going to jump into actually talking about the individual steps that take place during cellular respiration. What happens, what goes in, what comes out. So we're going to start here with glycolysis. And if we look at that term in and of itself, if you know some of the roots that are bound in that term, then hopefully the process itself makes sense. So when we look at this, right, glyco means sugar, lyse means to break. So we are taking glucose and we're going to break it into two molecules. So we're literally going to take the carbons and break it in half. So how is this going to happen? Well, when I look at glycolysis, if I really wanted to dig into all of the details, there's about 8, 9, 10 steps that are going on here. But we're going to break it down into two major pieces. So there's about 8 chemical reactions that get us from the start of glycolysis to the end. We're going to lump them into two phases. So when we look at this, I'm going to take glucose, which is a 6 carbon molecule, right? Which is what you're looking at here. Each of those C's are those carbons. And we're going to take that 6 carbon glucose and I'm literally going to break it in half. So we're going to split it right down the middle to create two molecules, 3 carbons apiece. And in order to do that, this process is going to require some energy, and then we're also going to get some energy back out of the overall process. So when I look at glucose, right, in order to break those bonds that are in that glucose molecule, I'm going to have to invest some energy. And so we're going to talk about this first phase of glycolysis here as our energy investment phase. And you should be labeling this in the diagram in your notes. And the reason we call it the energy investment phase is that that is exactly what I'm doing. If you invest in something, that means you put money somewhere to hopefully get out a final product. Well, in this case, we're investing energy. And so I'm going to take two ATP molecules and I'm going to break them down into two ADP molecules and use the energy of breaking that phosphate off in order to break this glucose in half. The second phase then is your energy payoff phase. And if you look at what's happening in the diagram here, hopefully that makes sense. So now I'm going to take 4 ADPs that are floating around in my cells, and I'm going to be able to phosphorylate them. I'm going to tack on phosphates and get 4 ATPs out of it based on the chemical reactions, the moving of electrons and hydrogens and all sorts of things that are going on in the chemical reactions that are happening here in this phase. So energy investment, I take 2 ATPs in order to help this set of chemical reactions go to break the glucose into two molecules, each with 3 carbons apiece. The energy payoff phase then, I'm going to take those two molecules, 3 carbons apiece, do some additional adjustments to them, move them around, move some electrons, move some hydrogens, and ultimately get these two molecules called pyruvates out of here. And each of these pyruvates also is a 3 carbon molecule. In doing that, it actually pays me back some ATP, 4 total, as well as to create the initial set of electron carriers that we're going to use for the electron transport chain later in this process. And we're going to see that every step along the way, we're going to create some of these electron carriers. And here, this electron carrier, we're going to take NAD plus, tack on some electrons to get rid of that charge, as well as tacking on a few hydrogens to create this thing called NADH. And again, that NADH electron carrier is going to get shuttled into our mitochondria and be used for the final step of cellular respiration when we talk about the electron transport chain. So just kind of tuck that away for now and keep that in the back of your head. So what I end up getting here is 4 ATPs out of my energy payoff. So when I look at this overall, glycolysis actually in and of itself nets 2 ATP molecules, right? It nets 2 ATP. And the reason we have to throw that term net on there is because although I get 4 out total, I put 2 in to begin with, right? So the net return on my investment, if you know any of that sort of business lingo, I should say. 4 ATPs out, 2 invested, and so I get a net profit of 2 ATPs that my cells can now go use somewhere else for some other process. Okay, so 2 investments of ATP, 4 payoff, and I end up netting, or I balance out 2 ATPs positive. I get 2 extra ATPs that I get to go use somewhere else. Now, here's the interesting thing, a couple of things to keep in mind when we talk about glycolysis as a whole. So, in terms of inputs, right, reactants, glucose goes in, ATP goes in, NADP and some phosphate goes in, and NAD plus goes in. So, glucose, ATP, NADP plus some phosphates here, and NAD plus. The reactants that come out are going to be these 2 pyruvate molecules, some ADP, some ATP, and NADH. Now, this whole process is actually taking place in the cytoplasm, and it is the only step of cellular respiration that doesn't involve oxygen. And so, technically speaking, glycolysis is an anaerobic process, right? Meaning no oxygen required. That's really going to be important. So tuck that away, because when we get to talking about fermentation, that idea is going to come back. So this is the only step of cellular respiration that does not require oxygen and does not take place in our mitochondria.
How do you turn that bite of food into a chemical that a cell can recognize and use as energy? The first step is altering the food into its component chemical compounds and then getting those molecules into your cells. That process is called digestion. Once inside your cells, the process of turning that bite of food into useful energy by cellular respiration begins. The process of digestion results with carbohydrates and other molecules being removed from the consumed food and transported into the bloodstream. From there, nutrients, like the carbohydrate glucose, will leave the bloodstream through a capillary wall and enter a tissue cell. Once inside the cell, cellular respiration will completely oxidize the glucose molecule, releasing high-energy electrons. The overall goal is to make ATP, a storage form of energy for most cells. Cellular respiration is a 4-stage process that begins with glycolysis. Glycolysis literally means splitting sugars, and it is the first step of cellular respiration, occurring in the cytoplasm of the cell. Glycolysis consists of 2 distinct phases, an energy investment phase and an energy harvesting phase. In the energy investment phase, 2 ATP molecules transfer energy to the glucose molecule, forming a 6-carbon sugar diphosphate molecule. This molecule splits, and the energy harvesting phase begins. During this phase, the 2 3-carbon molecules are converted to pyruvate, and ATP is formed. Glycolysis is a 10-step reaction that involves the activity of multiple enzymes and enzyme assistance. In the process, a net of 2 molecules of ATP, 2 molecules of pyruvate, and 2 high-energy electron-carrying molecules of NADH are produced. When oxygen is present, the pyruvate molecules and NADH enter the mitochondria, and the next stage of cellular respiration begins.
Okay, so once I've created those pyruvates, I'm now going to shuttle those pyruvates from the cytoplasm where glycolysis was taking place into my mitochondria. Here's the thing. In order to get that pyruvate to get across the mitochondrial membrane and into ultimately the matrix of our mitochondria, which is where our Krebs cycle is going to take place, I need to do some adjustments to that pyruvate molecule to A, get it into the mitochondria itself, and B, get it into a form that can actually get fed into my Krebs cycle. So in order for this to work, I've got to do some stuff to that pyruvate, and that's where this sort of half step that we're going to call grooming takes place. So grooming is going to take place in the cytoplasm as well as kind of across that mitochondrial membrane. and it's going to allow us to change those pyruvate molecules that came out of glycolysis into this thing called acetyl-CoA, or acetyl coenzyme A. So what's the point here? What's going to happen? So pyruvate's going to go in, but I need to attach this coenzyme A to help it get across the mitochondrial membrane, as well as to help feed it into the Krebs cycle, which is going to be step 2 of cellular respiration. But right now, carbon can make 4 bonds, and each of these 3 carbon molecules is all bound up. It's got as many bonds as it can make. It's attached to as many atoms as it can possibly hold on to. So in order to attach this coenzyme A to it, I have to create an empty bond. So what your cells do is take off this final carbon and create carbon dioxide out of it. So some of the carbon dioxide that you are breathing out right now, that is a waste product, if you will, of cellular respiration, comes from this grooming step. By removing that carbon, I now have an extra bond on this carbon to attach that coenzyme A to. Okay, so this carbon comes off, ends up being part of carbon dioxide that we breathe out. And now this blank bond here allows me to now attach that coenzyme A to it. Now there's a couple other chemical reactions and things that are happening within this arrow that once again I'm not kind of showing you because it's not vital to your understanding. Um, but it does mean that it's going to move some electrons around, move some hydrogens around again in the process of creating this acetyl-CoA, which also means I get another NADH out of here that I'm going to go use for that electron transport chain. So again, what goes in? Pyruvate goes in, right? That's coming from glycolysis. The NAD plus is going in as a reactant, and the coenzyme A goes in as a reactant. Products that come out of this, carbon dioxide, NADH, and acetyl-CoA. Okay? Now keep in mind that this particular diagram here is showing you for a single pyruvate. When we finished up glycolysis, we made 2 pyruvates. So be careful the way questions get worded and asked, because depending on how they are worded, they often will ask you, well, how many of this particular molecule get produced as a result of a single glucose? Which really means we have to double these numbers because I have 2 pyruvates that come off of glycolysis from that original single glucose molecule. So I get 2 molecules of carbon dioxide off for every glucose that goes in. I need 2 coenzyme A's for every glucose that goes in. I make 2 acetyl-CoAs for every glucose that goes in. I make 2 NADHs for every glucose that goes in. Okay, so just be aware of that, be careful of that.
The next stage of cellular respiration involves the movement of pyruvate into the mitochondria, where it undergoes oxidation. Each pyruvate molecule is converted into a compound called acetyl-CoA. In the process of pyruvate oxidation, electrons are transferred to NAD, producing NADH, and a carbon is lost, forming carbon dioxide.
Okay, so here is that grooming step one more time, right? Okay, so taking that pyruvate, removing the carbon dioxide, creating the NADH as a result, attaching the coenzyme A. Now that I have acetyl-CoA, and I'm now in the matrix of my mitochondria, and in the presence of oxygen, so now I'm in the matrix, I require oxygen for the rest of this to work, I'm now going to take a look at your Krebs cycle. Keep in mind that I mentioned to you before that the Krebs cycle can also be called the citric acid cycle, and the reason being is once that acetyl-CoA gets into this cycle, the first thing we produce is this citric acid or citrate. And so citric acid is where that other name comes from. So Krebs is the guy who discovered the cycle, and so he gets credit. So certain textbooks will call it the Krebs cycle. Citric acid's the first molecule that gets produced in the cycle, and so some textbooks call it the citric acid cycle. So just be aware that they can get used interchangeably, okay? All right, so again, this is taking place in the matrix of my mitochondria, and I now require oxygen for this to work. So for every acetyl-CoA that goes into this cycle, this is what happens. We take that citric acid. Again, there are a whole bunch of chemical reactions that are happening within this cycle that we're watering down and boiling down to the most basic components. We get some carbon dioxide out of here. So in the process of moving electrons and hydrogens and breaking down carbon, we're going to relieve the body, the cell, of some carbon dioxide. I will make some more electron carriers. So here's NADH here, here's some NADH here, here's some NADH here. So all of those NADHs will go to the electron transport chain. We'll also make a new electron carrier here called FADH2 from FAD+. We'll also get some ATP out, okay, as well as a little bit more carbon dioxide here as well. So when I look at this, okay, this is per pyruvate. So 2 carbon dioxides come out, 3 NADHs, 1 ATP, and 1 FADH2 are made per pyruvate. Now keep in mind that if I were to ask, or a question were to ask, how many of these molecules get made per glucose, that means this entire cycle has to go twice, right? One turn for the first pyruvate that came off, and a second turn for the second pyruvate, which means if I were to ask you these numbers per glucose, you'd have to double them. So 4 carbon dioxides, 6 NADHs, 2 ATPs, and 2 FADH2s per.
The next stage is the citric acid cycle, also called the Krebs cycle. Here, acetyl-CoA will bind with a starting compound called oxaloacetate, and through a series of enzymatic redox reactions, all carbons, hydrogens, and oxygens in pyruvate ultimately end up as carbon dioxide and water. The pathway is called a cycle because oxaloacetate is the starting and ending compound of the pathway. For every glucose that enters glycolysis, the cycle completes twice, once for each molecule of pyruvate that entered the mitochondria. During pyruvate oxidation and the citric acid cycle, a net of 8 NADH, 2 FADH2, 2 ATP, and 6 CO2 are produced for each glucose molecule. In order to understand how the majority of the energy is produced by aerobic respiration, we need to follow the NADH and FADH2 molecules to the next stage, the electron transport chain.